I'm going to show how to seal a sample on a microscope slide to prevent evaporation, allowing it to stay alive for several weeks at ambient conditions. I got this from David Walker's article in the September 2020 issue of Micscape, but I'm adding a couple of suggestions that I think might be helpful. If you're going to keep a slide going for a while, it's a good idea to write an identifier on it. I like to use the date, November 1st, for this one. Weighing the slide at each step lets you keep track of the amount of sample on it. Scales with a precision of 1 milligram have become very affordable. I find it convenient to write the weight on the slide as well. Add a little bit of algae and a drop of water from your favorite source. You may also want to add a trace of some nutrient. I usually use a bit of oatmeal. Not shown here but recommended, check the drop under the microscope to make sure you have any of the microbes you're interested in. Also, you may want to spread out the algae more than shown here. Weigh the slide again, write the result and calculate the sample weight, 53 milligrams here. Now smear a thin layer of Vaseline on a convenient surface. And scrape the edges of the cover slip across the Vaseline film to get the desired amount on each edge. Place the slip over the sample drop and tamp it down to spread the water over the whole area. Weigh the slide again and write the final weight on the slide, trying not to bump the camera stand. If the slide is stored under lighted conditions, the algae will generate oxygen for the microbes. However, too much light will result in formation of bubbles, degrading the view. You can assess water loss by weighing at intervals. Usually they're very stable after the first day. Subtracting the weight of the slide plus sample from slide alone gives the sample weight, 53 milligrams, which, assuming neutral density, is also the volume in microliters or cubic millimeters. Recording the weight of the whole assembly after placing the Vaseline-treated slip allows keeping track of any evaporation. In this case, after the first day, the weight is down by 13 milligrams, presumably from evaporation of water that squeezed out during placement of the slip. But after that, the weight stabilizes, indicating a final volume of about 37 microliters. With a standard 22 millimeter slip, and allowing 1 millimeter inside edges for Vaseline, this gives 20 by 20 equals 400 millimeters squared. Approximating 40 microliters or cubic millimeters dividing gives a sample depth of 0.1 millimeter or 100 microns. I've prepared a number of slides like this, varying the sample source, volume of water, amount of algae, added nutrient. Starting communities are extremely variable, some very diverse and some with just a few species. Most of the slides show these communities developing over several weeks, with some species proliferating and others dying off. The main reason for sealing the slides like this is to allow for long time-lapse videos without the need to add water to compensate for evaporation. I've done time lapses of rotifer and gastrotrich egg development that covered days. This is a long time to tie up a microscope, but there are plenty of subjects that benefit from shorter time lapses, a couple of hours to overnight. As noted earlier, too much algae and too much light may result in growth of oxygen bubbles that can interfere with viewing. I've found that the bubbles will usually stop growing and even shrink when the light intensity is sufficiently reduced. When I prepare a slide, I like to first make a quick time lapse of the full area to see a couple minutes compressed into a few seconds. This shows the motion of many rotifer-sized microbes and gives a sense of how many are there. These micro-ecosystems can stay alive and interesting for several weeks. This one is now at 8 weeks and still shows rotifers, a copepod, coleps, and paramecium bursaria. This slide was made 10 days ago and shows a lot of microbe motion at 30x speed. Close-ups show uroleptus, gastrotrich, euplotes with coleps, I'm not sure what these green spheres are. The oatmeal speck has had time to ferment and feed a number of smaller species. Note that most of these videos are in 3D if you use red-blue glasses. I find this very helpful in viewing microbes moving through the complex algae environments. The cardboard glasses are very cheap and can be cut to make the blue-red filter shown on top of the microscope lamp. Keeping the condenser at a low position gives the depth of field needed for good 3D viewing. I described this and another 3D viewing technique using cheap polarizing filters in detail in an earlier video. Link in the description.